How does the brain work? How do we think and imagine? How can we find cures for neurological disorders like Alzheimer's, depression, or Parkinson's? To answer questions like these, we need to understand how hundreds of billions of brain cells, or neurons, communicate normally, the cause for alteration, and blockages in neuronal communication and neurological diseases that lead to impaired emotional and cognitive functioning. Scientists in the past have attempted to understand how the brain works by externally stimulating specific brain regions by the use of electrodes that were attached to the subject's brain. They then studied the physical and emotional response created as a result of the stimulation. More recently, with the advent of the fMRI or functional magnetic resonance imaging in the 90s, scientists were able to measure brain activity and create images of activated brain regions non-invasively by using powerful magnets that detected changes in blood flow to the brain. The subjects were exposed to varying stimuli such as sounds, images, thoughts, and smell. Although these studies clarified the role of different brain regions and their respective responses to varying stimuli, these technologies lack the precision and the specificity necessary to understand how neurons normally communicate with one another and how impaired neuronal communication gives rise to neurological diseases and disorders. Enter optogenetics, a revolutionary technology that uses light to control genetically encoded light-activated proteins to selectively manipulate the activity of the targeted neuron without affecting other neurons in the brain. So how does optogenetics really work? The technology of optogenetics in the most basic sense is much like Facebook chat features. Just like how you can turn on Facebook chat and create groups to chat with some friends selectively from your entire friends list, with optogenetics, scientists can turn on specific neurons without affecting other neurons by photoactivating light-sensitive protein channel rhodopsin 2 that are genetically encoded into the targeted neurons from microbes using a method called gene therapy. Another chat feature on Facebook is the Mark as Offline feature, which allows the user to represent themselves as offline to specific individuals from their friends list. Similarly, another photoactivated light-sensitive protein acquired from the microbes is known as halorhodopsin, which can be used to inhibit or turn off specific neurons when shined with light. For example, when optogenetics, scientists can study how a specific subset of neurons contribute to a particular behavior. If we can turn on a set of neurons, we can figure out what behaviors are initiated by those neurons. Likewise, if we can turn off the same set of neurons, we can see how the behavior changes in the absence of those neurons. Granted so much control and specificity in experimentation, new drugs developed will target specific neurons that may be functionally impaired in neurological disorders with almost no side effects. Although optogenetics is not approved for human clinical trials so far, further studies and development in optogenetics techniques will likely present a solution for many life-threatening neurological disorders.